name is Sonam Choden. I am gonna do my final presentation. Polymerase chain reaction, also known as PCR, is a popular technique used for amplifying or making many copies of specific DNA segments from a mixture of DNA template molecules. It gives very precise results, and we used it to make multiple copies of beta-galactosidase. There are three steps in which PCR works. Number one is denaturation of the DNA template, in which the two DNA strands in the helix separates to form two single template strands. Number two is annealing of the primer. The DNA template specific primer attaches to 3' end and the 5' end of the specific template strand according to the bases found on the template. And number three is extension of the complementary DNA strand, also known as polymerization. The process repeats multiple times to produce many copies of the DNA. After that, the replicated DNA molecules are run through a gel electrophoresis to determine if the gene of interest is produced. The DNA strand is also run alongside DNA molecular ladder. This is used to estimate the size of the new DNA that was produced. The gel electrophoresis works because of the negative charge present on the phosphate group of the DNA molecule. When the agarose gel is provided with electricity and the DNA strand is submerged in it, the DNA strands will migrate toward the positive electrode of the electric fields according to their size. These sizes can be compared to the size on the ladder to determine the number of base pairs. Comparing the DNA ladder and the PCR product under blue light, it was seen that there are about 3,000 bases in each DNA strand of the PCR product. This confirms that the correct gene was produced because the gene template, which is beta-gal, contains 3,075 base pairs as well. The PCR product contains buffer components and other impurities like enzymes. Some impurities may alter the end of the cleaved DNA strand and reduce the yield of ligation. In order to prevent that and to get rid of all the unwanted molecules like salts, it is crucial to purify the PCR product before digestion. One of the PCR product purification methods is by running the product through an agarose gel. Agarose gel purification involves binding of the nucleic acids to a silica column, washing the impurities away with ethanol, drying it, and finally eluting the purified DNA fragments from the silica column. After the DNA has been purified, it needs to be digested by restriction enzymes into fragments so that the DNA can be inserted into a compatible plasmid. The restriction enzymes, just like the other enzymes, bind to the DNA molecules by matching their shape to that of the DNA molecules and breaks the DNA helix into fragments. Each restriction enzyme only works for a particular nucleotide sequence. Restriction enzymes either cut straight through the DNA strands, creating DNA fragments with blunt ends, or at different positions, resulting in fragments with overhanging nucleotides, also known as sticky ends. This sticky end makes it very easy to bond with other DNA fragments. Two restriction enzymes used in this digestion were HINT3 and BAM2. They were used along with extra flanking bases, which holds the restriction enzymes in place. Ligation is the process of binding two nucleic acid fragments together with the use of enzyme known as ligase. Ligases catalyze the joining of the phosphate group sticking out the sticky end from the 5' end of the DNA strand and the hydroxyl group sticking out from the 3' end of another DNA strand. The plasmid and the gene that goes into the plasmid must be cut with the same restriction enzyme for the plasmid and the gene of interest to have complementary sticky ends. After ligation, this recombinant plasmid is transferred into bacterial cells through transformation. Heat shocking them opens pores in the bacterial membranes and makes it easy for the plasmids to go in. The plasmids used in cloning contain an antibiotic resistance gene. With this, the cells can be checked to see if they have taken in the plasmid with the gene, and from there, only the cells with the plasmid and the gene can be grown in bulk. The cells in the plate grew and turned out to look like the picture shown on the left. It has some colonies that can be screened for the specific gene of interest, which is beta-gal. After we ran a gel electrophoresis, the result shows that the PCR colony contains beta-gal. After confirming the gene of interest in the PCR colony, the cells undergo induction. Induction in the cell is a process where the genes in the cell have to be activated by certain environmental factors, and when they are activated, the desired protein is made. By controlling induction, a gene-containing cell can be used to mass-produce a certain type of protein. The special cell line, known as E. coli BL21, is what this experiment used for the induction. These cells are also very easy to use when something needs to be expressed through the lac-operon. Lac-operon uses a non-hydrolyzable analog of lactose, known as IPTG, and the T7 RNA polymerase, which is present in it, transcribes for beta-galactosidase gene, inducing its mass production. After affirming mass production of beta-gal, the cells were lysed by freeze-thaw method. While freezing, the iced crystals are able to puncture the membrane and the cell contents are released, making a viscous solution. The long DNA strands are chopped up and precipitated and collected in the form of protein pellets. Liquid chromatography is the technique to purify specific proteins according to their specific attributes or properties such as charge, size, hydrophilicity or hydrophobicity, etc. It has two phases, liquid mobile phase and solid stationary phase. The liquid mobile phase dissolves the sample, and the solid stationary phase is present in columns which contains beads. The beads are manipulated to exploit the protein characteristics to purify them. To for example, ion exchange chromatography and size exclusion chromatography uses two different characteristics to purify the proteins. Ion exchange chromatography, also known as IEC, separates the proteins based on their charge. For example, beads are positively charged to purify the negatively charged proteins, and vice versa. Beta-gal is negatively charged, so it uses anion exchange as it is negatively charged. In size exclusion chromatography, the molecules of the sample are separated according to their size, where larger molecules are eluted first. The beads in the column have openings in them that trap the small molecules, making it hard for them to flow smoothly, while the larger molecules go around the beads, which leads to faster flow. Then the samples collected from liquid chromatography are run through a STS page. It separates proteins according to their size and works very similar to the gel electrophoresis and determines if the sample is the desired gene. Results show that we obtained the desired beta-gal as the sample beta-gal has a weight of 114 kilodalton, and the beta-gal produced from our experiment also has a band right above the 100 kilodaltons. BCA assay method is used to determine protein concentration via change in color. The change in color is induced by reduction of copper 2 plus to copper 1 plus and its chelation. This purple color of the protein can be quantified by running it through a UV vis at 562 nanometer, which gives an absorption curve. The curve is compared to a standard curve given by a protein of known concentration, such as bovine serum albumin (BSA). The beta-gal concentration was found to be 1.630247 mg per ml. Activity assay is used to see if the protein we produced is working or functioning properly. It makes use of the reaction that the particular enzyme catalyzes to affirm the activity of the enzyme. In this experiment, we determined if the beta-gal prepared and purified in the previous experiments was functional. Beta-gal hydrolyzes lactose to glucose and galactosidase. However, these products are colorless and do not show in the spectrophotometer. We used ONPG, a derivative of lactose instead of lactose. Mixing it with the bitter gal turned the solution yellow, which indicated a formation of O-nitrophenol, meaning the enzyme was functional. The spectrophotometer data also showed absorption at 420 nanometers, confirming the result. The control, which only contained ONPG and no beta-gal, neither changed color nor showed absorption. After determining that the protein is active, kinetics assay determines how active a protein is. It is done by gradually inhibiting the reaction by adding more and more inhibitors. The inhibitors can be either competitive or non-competitive. Michaelis-Menten kinetics can be used to analyze the kinetics of the reaction. In case of beta-gal, we can use IPTG as an inhibitor to see how the reaction rate changes. Thank you.